Welcome to today's webinar on PDBe KB Aggregated Views. I am Preeti Chaudhary, a developer from PDBe Europe team. Like as Jay said, feel free to put your questions in the Q&A box at any time. I'm also joined today by David Armstrong, a database curation expert at PDBe. And between us, hopefully, we should be able to answer all your questions. Of course, you can reach out to us at any time via email or our social media channels. I'm talking today from PDBe in Europe, placed just outside Cambridge in UK. We are one of the four. One of the four core PDBe archive members, along with RCSP PDBe and BMRB in America and PDBJ in Japan. And as such, we help to collect, curate, store, and distribute the atomic structural informations to the users all over the world. Talking about the atomic structures, when we look at a 3D coordinate file, we start with what is this? 3D coordinates by themselves, they are just shapes and they are not of any intrinsic biological value unless we add on top of it some additional information. For example, in this particular protein, you can see it's a SARS-CoV-2 main proteases, and it leaves the coronavirus polyprotein at various sites. It exists as a homodimer, as you can see. Talking about more about its biological context, you can see over here its catalytic residues highlighted and ligand binding residues also highlighted. Such value-added annotations give scientific meaning to 3D coordinates. With PDBe knowledge base, we would like to collate such value-added annotations. Now, PDBe KB is a community-driven resource for structural and functional annotations. It was established in 2018. As a core member, we create data standards, we create data access mechanisms, and help to reduce fragmentation of the various annotation data. And the aim of our PDBe KB is to create one-stop shop for the users. If you look at talking about fragmentation of the data, often you see that multiple structures for a single protein are often solved in various crystal conditions, experimental conditions. Usually, these structures correspond to a fragment of a portion or a portion of a protein. And there are tons of specialist databases out there which have information with respect to each of these PDBe entries and various types of information. However, this data is fragmented and does not follow FAIR principles. It is very difficult to find them and transfer all these annotations on a protein level. The aim of PDBe KB is to integrate and interconnect all these annotations in order to provide a comprehensive aggregative view of on a more biologically meaningful entry, in this case, full length of proteins. Meanwhile, following FAIR data principles, which make it the data easy to find, accessible, interoperable, and reusable. In PDBe KB, we have data from various resources covering the domain annotation, R-Flum classification, ligand binding sites, macromolecular interaction, variants, sequence conservation, and many more. If you look at the overall infrastructure of PDBe KB at the moment, we have 29 consortium members. And all these members provide their annotations in form of JSON format, which every week is handled by our weekly data validation and integration process. This parses these files and stores the data in a graph database. This graph database is available for users to download. This is also used to expose the data at various aggregated endpoint, and this is and via this users can programmatically access this data. We also created PDBe KB Aggregated Views, which are a set of web pages, and in this talk we will focus on these web pages. with respect to aggregated view of proteins. Along with aggregated view of proteins, we also are developing aggregated view of ligands, complexes, domains, and other entities. And together with all these aggregated views, the aim is to change the structural context of the data and add biological value or insight into the structural data. But before talking about more about aggregated pages, let's see how you can access them. So these protein-centric PDBe KB pages can be accessed via a PDBe identifier or a UniProt accession code. You can also go to these pages from our PDBe entry pages, from our search, or from UniProt entry pages. We also have a PDBe KB protein aggregated view landing page, and you can access your protein of interest from there as well. Now, let's look into the aggregated view of proteins in details. We'll start first with the home page, which shows the summary of the protein. Here you can see a brief view of the summary page. This essentially gives you the name of the protein, as in this case, spike glycoprotein, from which organism it is, UniProt accession, and all the other biological functions involved in this particular protein. Sometimes you can also see the EC number and disease associated with that particular protein, wherever it is known in UniProt database. The second main feature of this page is the 3D viewer MolStar, which shows you a representative PDBe chain covering various segments of the protein. Here the currently displayed PDBe chain is highlighted blue over here, and you can see the details of the PDBe chain. You can see the UniProt residue range, which it covers along with its coverage in percentage. For this particular PDBe entry, you can go to PDBe entry page from this button. Also, to know more about the details, how to navigate and go to other different regions of the sequence, and how we select our representative chain, you can find this information in the help text in the tooltip. In general, we use the PDBe entry with highest number of observed residue from various regions of UniProt protein sequences, and you can navigate from these arrows by clicking on these arrows on the next region or the next segment of your protein. Here you can see the structure for the next protein. This is essentially an NMR structure, which you can see the different models, and you can click on these arrows to navigate the various models for this particular structure. Next, moving on, we also have a feature called superimposition. Every week we run superposition pipeline, and here each PDBe chain is structurally aligned using Gazamt software, and these structures are clustered together based on structural similarities. You can view these clusters for a given region from this particular by clicking this button. It will open a 3D viewer MolStar showing you the superposed structures for that region. You can also click on the button which is present below the structures icon. This again will open the 3D viewer of the MolStar showing all the superposed structures for any given region. To read more about the superposition pipeline, you can refer to this link. Moving on clicking on the button for superposition, you see the MolStar window. Here you see the different clusters. For example, for this particular spike protein, we have 7 different clusters for the segment 1, which ranges from residue 319 to 591. Each of these clusters correspond to a uniform structural conformation. These may or may not have biological relevance. By default, you only see the representative PDBe chain. However, you can unhide the other chains by clicking this I button. You can also navigate into various segments by choosing this drop-down menu and selecting the corresponding region of your interest. All the data for superimposition is available over the link over here. You can find the rotation matrix and the clustering dendrograms at this particular link. We also have, while superimposition, we also align any bound ligand within the cluster. And this view you can find by clicking the button present below the ligand icon. This is called 3D view of superposed ligands. This opens a 3D viewer showing you the superposed ligands. Now, this view is very helpful in highlighting all the common ligand binding site residues. On the right-hand side, you see the ligand and the various carbohydrates which are bound to your PDB chains. You can unhide any of the ligand which is not of your interest and change representation using these action buttons. Moving on, you also have these icons, which gives you a quick overview of the structural data available for your protein of interest. For example, total number of structures, total number of ligand bounds, how many macromolecular interactions are there, and similar proteins and various publications. You can download all this data by clicking on this download button. Present below each of these icons. And here you can download files like MMSIF file, FASTA files, and even validation data. For many, in case of polyproteins, as the spike protein over here, you will also see towards the end a section called processed proteins. This essentially shows you a list of all the processed proteins which can be accessed, and they have their separate aggregated view, which can be queried using the pro-identifiers from UniProt. For each of the spike protein, processed protein, you can click on this segment, green colored box, to view the structure in 3D in MolStar. And you have these icons showing you the quick glance of available structural data. For instance, number of PDB structures, number of PDB ligands, and number of interacting partners. And you can also view the dedicated processed protein page using this button over here. Between all these various sections, you can use the navigation bar and click on the corresponding tabs to navigate between various pages. Alternatively, you can click on each of these icons to view the relevant page. Now, let's talk about ProtVista, a component which is heavily used throughout the PDBe KB aggregated views. In this, ProtVista is essentially a sequence feature viewer, which was jointly developed by UniProt, PDBe, Interpro at EMBL-EBI. And this is freely available for anyone to use at GitHub at this link. Now, here you can see in this viewer, this is the protein sequence, a scale from 1 to the total length of the protein. You have ProtVista main component or main section and the subsection which shows you annotations for various residues. You can collapse by clicking the arrow or the main ProtVista section. If you hover over any region or click on its details, you can see the details of that particular residue on the tooltip. There are icons like this circle where you can focus on a particular subtrack, and you can hide a particular subtrack using this cross button. In ProtVista, you will see evidence-based section in blue color, and you will see the prediction-based section in gray color. You have a reset button, which resets the zooming and the hidden tracks. and takes you to the default view. You have a section for navigating the range where you can put or highlight a specific region of your interest. If you highlight it to high level, you can even see the amino acids. You can use your mouse scroll as well in order to zoom. You have a special button for hiding the various sections. For example, here I have hidden PDB structures and secondary structures. So now let's talk about the structures section information in aggregated view of proteins. This is how the structure page looks like for a given protein. Here we are looking at spike glycoprotein. It mainly has two sections. First, you have the 3D viewer showing the PDB structure of the protein. And then you have the ProtVista sequence viewer, which shows you all the structures and various other information. You can see by default, the structure which is first listed in the ProtVista is highlighted in the MolStar viewer. And if you want to view the specific PDB entry page, you can click on this button. In order to see, you can click on any segment which you want to see in the MolStar viewer over here, and the structure will be displayed. For example, over here, if I click at this section, it highlights 7SO9. Then in this section, you see at the top experimentally stalled structures which we have in PDB. Then we also have data for predicted models from various resources like Swiss model, SASDB, and also AlphaFold-DB. This section gives you an idea of the region for which we have the known structure. In general, the PDB entries are sorted by maximum number of observed residues and resolution. If you look at the details for each of the subtrack in PDB structures, you will see there are links and the button is highlighted as green if it is involved in a complex or if it has interactions with DNA and RNA. In this case, there is no structure which is bound to DNA and RNA, hence it is grayed out. You can also see the link to various structures to which any ligand is bound. Towards the end, you see the resolution of that structure. In case of polyproteins, you also have processed proteins, and you can see the location of various processed proteins, where they are chopped off in the polyprotein. And this view enables a quick identification and comparison of various processed protein structures. You can also see the secondary structure elements which are observed for a representative PDB chain for each of your covered region. You can also see domain information. We have domain information from PFAM, CAT, SCOP, and even Interpro, which is highlighted in this section. In terms of flexibility, we have data coming from MobiDB, which basically gives a consensus disorder prediction from UniProt sequence for a given UniProt sequence. For more of the details about our other partners, we will talk about it in annotation section. You can view the 3D view of superposed structures as discussed in the summary section from this place as well. It will open a 3D viewer showing you all the superposed structure for any given region. You can download the data by clicking on this button for all the structures. For example, in this case, I can download all the 890 PDB entries corresponding to this particular UniProt accession by the click of single button. And I can choose MMSIF file, FASTA files, or even validation data. All the annotations which you see in ProtVista viewer can also be downloaded in CSV format by the use of annotation button. Moving on to the next section, which is the ligands information on aggregated view of proteins. This is how the ligand page looks like. This section shows you all the ligands. By default, it only shows the ligands which are directly bound to the protein of your interest. However, if you click on this button, you can see all the ligands which may or may not be directly interacting with your protein. In this case, the directly interacting ligands and all ligands are of same. All the ligands are interacting, and hence you see the number is same. You can filter your ligands list or gallery using molecule name or PDB ID or even the 3-letter code of the ligand. We highlight the ligands with known functional annotations in green, and the data for them is shown over here at first. These annotations can vary from cofactor, which is coming from our cofactor pipeline. We also map to drug bank and RIA database in order to annotate ligands as drug and reactant-like respectively. If you hover over the head code, you can see the full molecule name. Over here, you can see found into PDB entries, and this links gives you PDB search results listing all these PDB entries. Or each of this button, each of this image in the ligand gallery is essentially linked to 3D visualization. On clicking on the ligand image, you will see a MolStar window opening, showing you the ligand binding pocket. If there is more than one ligand which is bound in more than one entry, you have the drop-down menu and you can navigate between various PDB entries. You can click on this download button in order to download data for this particular ligand. Now, the next part of this section is ligand binding residues, and this is shown in the ProtVista viewer, and it shows you the residues which are involved in binding. Here, this view can help you identify the commonly interacting residues. These are we usually color code them, or there is a color gradient, and mostly the more consistent the interaction is, more darker it is in color, and hence it can allow a user to have a clear identification of key interacting residues. If you hover over a residue in the tooltip, you can see the residues details and the entries in which they are present. If you collapse the ligand binding site, often you can see the overall ligand binding residue overview. And this collapse view can also help in finding patterns in binding events. For example, over here, you can see these particular two ligands have similar binding sites and they have same scaffold. And you can also identify the common binding residues. You can download the data using, again, the download buttons and also the ligand annotations. The 3D viewer shows you the superposed ligands as discussed in the summary section. The same view is accessible. You can download the structure data. So here, only the structures which are ligand bound, you can down them specifically, all kinds of information, and all the ligand related annotations which you see in ProtVista viewer can be downloaded using the download button over here, ligand annotations. Moving on to the aggregated view of proteins, you have molecular interaction information. This is how the interaction page looks like, and this section gives you the details about the macromolecular molecules which interact with protein of your interest. Here you can see that there are 17 unique interacting partners. You can filter these results by molecule name, PDB code or PDB ID or UniProt accession. If you hover over them, you can see the full name of the protein. And here this link takes you to all the relevant entries in which the interacting partner is this particular protein with your protein of interest. You can view the specific protein page for the interacting partner by clicking on View Protein Page button. Similar to the ligand section over here as well, each image in the gallery is linked to 3D visualization, clicking on which you will see a small star window showing you the interacting partner and your protein of interest. If a molecular interaction partner is present in more than one entry, you can use this drop-down button and navigate between different entries. Moving on, the next part takes you to the ProtVista viewer, showing you the interface residues which are involved in macromolecular interactions. You can see all the residues which are involved at the macromolecular interaction interface. Usually the homomeric interfaces like this, the self interactions are shaded in darker shade of blue. If you hover over any residue, the details of the residue are highlighted in the tooltip, and it gives you the details about how many entries it is present as well. If you collapse the interface interaction, you can see the overview of common interface residues which are involved in interacting with macromolecule in general. Then you can download the structural data, all the complexes. For example, in this particular case, you have 473 PDB entries which are interacting with macromolecules, and you can download various type of data using this button, download button. All the annotations which you see in ProtVista viewer for interface annotation, you can use this button to get those in CSV format. Moving on, now let's talk about the functional annotations information in aggregated view of proteins. Here you see a ProtVista viewer which shows you predicted and evidence-based annotations in gray and blue respectively from various PDBe KB resources. So this annotation section has all the functional annotations coming from our different PDBe KB partners. In order to know the details about each PDBe KB partner, you can hover over the resource name and it will tell you the details. For example, over here you see Metal PDB is a database of metal sites in biological macromolecular structures, and similarly you can look at the details about other resources while hovering. If you hover over a residue, you can also see the annotation details in the tooltips. For example, over here, the cysteine 1732 is coming from metal PDB, and it says that it is a metal binding site. Then you also have domain mappings, ligand binding sites, and interfaces over here. And this allows you to compare all the annotations from resources from what you see in experimental data or what is seen in structures. We also have information on sequence conservation coming for calculated for the entire UniProtKB. This is how the zoomed in version of the sequence conservation view looks like. If you zoom in the details, you can order the residues based on amino acid property or probability. The multiple sequence alignment file can also be downloaded using this button. If you collapse the sequence conservation view, you can see the high level details on the conserved residue. And if you hover over it in the tooltip, you can also see the conservation score. We also have variant information in variation section, which gives you details about various sequence variants on a protein function and their relation to diseases. You can filter based on the variant effect and their origin. In this tab, you can download all these annotations which you see over here using your annotation download button in a CSV format. Moving on to the similar proteins or similarity information on these aggregated view of proteins. This is how a similarity page looks like. Here you see proteins with 90% or more sequence identity to your protein of interest. You can see structures which have structural data. And you can see structures or similar proteins without any structural data, which is essentially the sequences coming from UniRef 90. For each of these similar proteins, you can see the coverage of the representative structure, and also you can see what is the general structural data available in terms of number of structures or number of ligands and interacting partners, respectively, from these icons. If you click on this blue box over here, or you can load the structure in the mol star over here, and you can go to the specific PDBe entry page from this go to PDBe button, and the corresponding protein page for the that particular similar protein can be viewed by clicking on this button. This list of similar proteins can be filtered based on species, taxonomy, and protein name. You can expand by clicking on this icon, which will give you a full list of the similar proteins. Now, moving on to the publication information in aggregated view of proteins. This is how the publication looks like. You can see there are three main sections. The first one being primary publications from PDBe entries related to protein of your interest. Then you have reviews where you see the PDBe entries related to protein of your interest are mentioned. And lastly, you see the publication from UniProt for the protein of interest. You can filter these publications based on PDBe ID or PubMed ID, or even the keyword in the title. You can navigate between these pages using these arrows or numbered buttons. For each of these publications, you will find link to EuroPMC and DOI links. And all the PDBs which are mentioned in that particular article or publication, there are links also available straight away over here. Now, apart from these views, the additional information which we have, all of these data which you saw on PDBe KB Aggregated Views, you can access programmatically via our API endpoints, and here are the links. This is the link for the tutorial and videos on using those API endpoints. Like I mentioned before, you can download the database using the Neo4j database, which powers these aggregated view in-house, and query that database as per requirement. All the components which you see on aggregated views, they are openly available on GitHub at this link, and anyone is free to reuse.
So we have one question already that I've marked to answer here, but yeah, if you do have any questions at all, please add them to the Q&A and we can answer those now. So yeah, the first question, Preeti, is from Carol Burker. And he asks, how do you determine what ligand is a cofactor and which is not?
Okay, thanks, Carol, for your question. So in order to define what is a cofactor or not, we have a cofactor pipeline in place, which runs every week. And this was the work which was done in collaboration with Janet Thornton's group. And we use parity score in order to match the existing ligand to the set of list of known cofactors, and that is how we define cofactors. And for more details, I can link you to the paper as well, I think, which shares more details about this cofactor pipeline of ours.
Okay, thank you, Preeti. So we do have another question that's just come in, which is from Ayla. Is there any way which we can introduce one amino acid mutation and compare it with the wild type?
You mean, thanks, Ayla, for your question. I am not so sure if I understand correctly, but directly on the pages, you can, so from the pages you can see, for example, if you go, I should have highlighted in my ProtVista viewers, but if you go in my slides, but if you go to the ProtVista viewer, we have mutations highlighted in orange color in the ProtVista viewer. And there you can see directly what is the experimentally, what is the mutation known, what is the already known mutation which someone has solved a structure, with already a mutated structure. I would, so that's how you can see in the archive the list of all the mutated structures. But in order to, if you want to introduce a mutation in a structure which is not seen, or it is not, we don't have a crystal structure solved for it, you can use viewers, or you can use 3D visualization tools like PyMol or even VMD to introduce the single residue mutation. However, you would need to do some kind of energy minimization or stabilization in order to get the real effect. Or you can use a tool in order to mutate your structures, which there are tools as well to introduce mutation in the tool. But on the aggregated view of proteins, you can see only the mutations which are seen in an experimental structure. I hope that answers your question.
Yeah, just as one little additional point is one of the data sets that we do integrate into our annotations is FoldX, who do this for some examples. So if there are examples that FoldX have done this mutation and then predicted how this might affect the structure, then again, those are linked. Otherwise, yeah, everything that Preeti said was spot on. Okay, so we have another couple of questions come in. So there's a question from Rahul here. When we query a specific protein, is it possible to see the list of proteins within the same family in form of a dendrogram or something similar to have an idea about proteins in the same family sharing a percentage sequence similarity or percentage structural similarity?
Thanks, Rahul, for your question. At the moment, you can only filter based on taxonomy using the search field, which I showed in one of my slides. But among those, you cannot, at the moment, we don't have exact sequence identity associated with it. But all the structures which you see are generally more than 90 or more than 90 sequence identity. We are looking to expand, and at some point we would like to include sequence similarities, proteins with sequence identity greater than 75%, essentially taking sequences from UniRef 70. And I think at that point it would be nice to maybe have a similar cutoff or a selection parameter, which essentially can give a user to choose between a certain sequence similarity or identity range. But at the moment we do not show exactly exact value of similarity.
Thank you, Preeti. We have a question here which isn't necessarily focused on PDBe KB, but you maybe talk about how we present sequence information at KB. The question is, how do we determine the sequence of amino acids in a particular protein, like, for example, COVID-19 viruses? So I guess what we can't deal here with necessarily the experimental side of things. Obviously, what we have is the data.
Yeah.
the sequence information has already been determined in other databases, for example, UniProt. We obviously, through the ProtVista viewer that Preeti showed, we kind of display this sequence information, so you can view it for a particular UniProt entry. But yeah, this isn't really related quite to what we display at PDBe KB.
Yeah, but you can search with respect to the organism name if that's what you're looking for. So, for example, you can search even on the PDBe KB page, or even using our search page. You can use the taxonomic ID and then look at all the already known PDBe structures, and then have a look at their amino acids, and then from there you can go to similar proteins and then look at their sequences of amino acid to have an idea of what your protein of interest might look like.
Great, thank you, Preeti. So there's another question here from Rahul. For the interaction data annotated here, does PDBe KB also pull interaction data from BioGrid?
Thanks, Rahul. So at the moment, what you see in interaction section is the data the interactions which you see in the crystal structures. So that is what you see, the data in the ProtVista viewer, which you see in the interface at the interaction section. At the moment, we are not pulling the data from BioGrid, but it's just the experimentally observed data. You can also have a look at all our partners at pdbekb.org/partners to have a list, idea or full list of all the partners from which our page we are taking data. And in case if you are interested in submitting the data to us as well, you have guidelines also mentioned on that page.
Excellent, thank you, Preeti. I hope that. So just to follow up on that from Rahul, so it doesn't have any predicted interactions, and can we say that the annotated experimental data is of high specificity? Is that correct?
So you see the functional annotation part, you see in the annotation section, you see all the data which can either be from experimentally known, or it can be a prediction, and both of them are coming from the partner resource. For particularly at interface interaction, we don't have this data coming from the partner resource, but only the experimental, what you see in the structure, that's what is listed. But in future, if we add a resource with predicted interactions, I think that would be available you can see that in functional annotations.
So just having a quick look, I think we do in the annotation section, we do integrate data from 3D complex. That's curated, that's not actually predicted, that's curated interface annotations. But yeah, everything that's on the interactions page specifically is all experimental, and it's all calculated using our software, PDBe that assesses those structures and where those interactions are. So as you've suggested, of high specificity, they are experimentally determined, and there is validation to determine how good those structures are. So again, you can validate that against what's there.